coordinators of this GujBT e-lecture series, Dr. Ram Sudhar and Dr. Kamlesh sir. So today's topic is cryopreservation. It is one of the important topic of plant tissue culture or plant biotechnology. So as the term indicates, it is one of the preservation techniques, and mostly the plant parts or the resurrection plant and the other. So in this case, we will discuss little about what is cryopreservation, what are the different steps, what are the protocols, what are the advantages and disadvantages. So initially, we will see these many topics, the contents of this one. We will discuss about what is this, the background of this one, then what might be the different choice of materials or which materials can be utilized for this process, and what are the prominent steps involved, and naturally its applications. And further, I will deal with these two which are connected to cryopreservation. Okay, and some of the examples of these many banks you can say, and the respective references we will discuss. And time to time, I will highlight which points are important regarding the cryopreservation are concerned or your exam point of view. So let us start with the definition of it. So once you try to mention about this word is actually ultraphasing, you can see. This is ultraphasing. Ultraphasing, but importantly, not only it will freeze the material, but those samples which are being stored over here, those were in viable condition. That means it can be reused further after having a storage period. And importantly, the temperature is around minus 196 degrees Celsius. In a regular freezing condition, generally 4 degrees Celsius is generally maintained. In deep freeze condition, minus 20 degrees Celsius, which were day-to-day life we are utilizing for storing the ice cream type of thing. So this cryopreservation, why there is a need of this minus 196 degrees Celsius? Okay, because where the metabolism is actually totally being stopped, virtually halted. At that time, once it is being incorporated into this temperature, all the metabolic processes, whether anabolic or metabolic, that will be stopped over there, virtually halted over there. So let us go through the history part of it, where it was being started, when it was being utilized, why it is being utilized. So it was initially started in the mid part of the 20th century. Okay, and mostly this cryopreservation is generally utilizing. Why they are trying to go for this one? They will try to store the different substances, including the animal sperms or embryos or cells or microorganisms, which are required further in the due course. So keeping it in mind that they wanted such type of gene banks, for that reason they go for this cryopreservation technique. So there are three different examples I have quoted over here. First one quoted, that is Sakai in 1960. He preserved the certain woody plants in cryopreservation condition. Secondly, the other example is cell suspension can be preserved by cryopreservation technique by Quattreno in the 1968. And thirdly, Nag and Street, they preserve the somatic embryos in cryopreserved condition. So these are the prominent examples which you should remember. Okay. Then why there is need of this? Why to do it? Under this, there are several features. First, there will be the important part is that there is the population crisis. Okay, huge amount of population is going on, and it is per second you can not imagine crores of new generation is being incorporated into this world. So this pressure, as well as what would happen, the food requirement is there, and to occupy this population we have to require these land resources. So what would happen, whatever lands and forests are being used, that means those plant materials were devastated. So for that reason, what would happen, there is need of the medicinal and aromatic plant species to be preserved, otherwise they were in the verge of extinction. This is the main motto, to preserve these requirements, okay, the various countries. Okay. So for that reason, some animal species also being in the or under the condition of the extinction. So to conserve this germplasm, whether the animal species, whether the plant species, there is a requirement of this technique, that is cryopreservation. So for this reason, cryopreservation is being the utmost important. What might be the materials used in the case of the cryopreservation? The first one is, the important one is, that is liquid nitrogen. Liquid nitrogen is used. At times, dry ice can also be used. But which one is preferable? Liquid nitrogen is preferable. Why liquid nitrogen? Because of these many features. What are the features? First one is, it is generally not reactive in nature, chemically inert in nature, and naturally the cost factor is also being essential. So it is low cost in nature, not toxic. Not toxic means if it is being utilized in for that conserved tissue, it will not react with that. So it is not toxic to that. As well as, this is also a good point, non-flammable. There is no need of any uh, fire to it, so it is a good option. As well as, it can be readily available. It is mostly found in wherever cryopreservation is needed, blood bank type of thing, they also require this. Okay. Now come to the steps. There are as much as 7 different steps are there. The first one is, you have to choose which plant material to be used under this one. That we will discuss later on. Okay. And before going for this, there is a stage called as pre-growth. Kindly switch, mute your mic. Kindly mute your mic. Shukla, Halak Shukla, Halak Shukla. Yes. So pre-growth is the second stage. Before going for preservation, you have to go for certain conditions that will be utilized in growth. And most important that, if you wanted to put this material in the cryopreservation condition, you have to add certain cryoprotectants so that those preservatives cannot incorporate into the plant material, and that is a must. So what might be those? There will be certain vitrification procedures are there. Some of the dehydration procedures are also there, and important one is encapsulation and dehydration. We will discuss that in details. Next one is freezing, that is the fourth one. Fifth one is storage. Under freezing, we will discuss these three parts. Then after storage, if you want to collect it back and or reuse, you have to go for thawing or reverse process, you can say. And then we have to identify which cells are survive and which cells are dead in nature. That also we will discuss. So under this, these three techniques were being utilized. Mostly question asked from these three parts: TTC method of staining, Evans blue staining, and FDA staining. So let us start with the first one, that is selection of plant material. So mostly, depending upon the stage of the plant, if you are going to preserve, then you have to decide that at which stage of the plant we should take those plant materials for preservation, or which the best factor is the plant must be a healthy one. Okay, whichever tissue we are going to take in for this preservation, the plant must be a healthy status. Okay, and importantly, which cells can be taken, this one is important. It should be meristematic. Here I would like to mention that meristematic cells are the important aspect of meristematic cells are they have the power of division and redivision. They have the power of division and redivision. So while storage, if you are going to store the meristematic cells, then it is a best option, or once it is being removed from this cryopreservation condition, they can able to further redivide. This is the first important feature of meristematic cell. Second important feature is it is living in nature. No doubt, you cannot go for the non-living plant materials. Okay. Third one is living means it should have nucleus. It should have nucleus, as well as in between cells or meristematic tissue. If you have taken, then there will be chances of the intercellular space. Okay. Along with these meristematic cells, this must be small enough. Naturally, meristematic cells are young in nature and it is rich in cytoplasm, so that it can able to survive or able to go for the further growth. Okay, so these cells can survive better than this part, highly vacuolated. So better you have to choose these meristematic cells as your plant material. Okay. And if you want to go for the further after selection of these cells, let us say you have prepared the callus. Those callus, particularly in case of tropical plant, preferable. Why? Because they are more resistant for freezing damage. Okay, because you are incorporated at minus 196 degrees Celsius, they have the capability to resist that. Otherwise, it would not be stored over there. Okay, and the important thing is that the cells do not possess water, so that it can able to go for the preservation technique. Okay, should be low freezable water. Tissues can withstand extremely low temperature. So these conditions must be fulfilled for that plant materials. If these conditions fulfilled, then definitely it can be go for the cryopreservation technique. Now, before going for cryopreservation, the second point is pre-growth. In case of pre-growth, what would happen? The treatment can be given to those tissue or plant tissues so that it can handle this liquid nitrogen further. So for that reason, the treatment must be given in a proper way. Okay. What does it mean? That means certain additives were being utilized so that it can able to fill the stress of this liquid nitrogen. Okay. And they have that ability to tolerate those conditions, for which these additives may be of these many types. Either it may be the stress hormone, that is abscisic acid, stress amino acid, that is proline, or an osmolyte type of thing, that is trihalose. Here I would like to mention that the proline is a closed imino ring structure. Proline has a closed immunoring structure. That's why during water stress of the plant, these amino acids are prevalent. And what the main point of this one is, as because it is having this closed amino group, it could not be metabolized very quickly. It could not be metabolized quickly. For that reason, mostly the prolines are found in case of or during the water stress condition. So for which these additives were utilized in that plant tissue so that they can able to survive in this stress condition, because you are incorporated in certain things which are not natural to the plant tissue. Okay. In some cases, partial tissue dehydration can also be utilized by the application of certain osmotically active compounds. Uh, Osmotically active compounds, let us say, in case of Catharanthus roseus, this is in Gujarat, it is called as Barmasi plant. And these cells were being taken by incorporating a osmolyte, that is sorbitol. Sorbitol before the freezing. Another example given over here, that is digitalis cells. It is being utilized 6% of mannitol. So let us say in exam, if question may ask, what are the different osmolytes utilized during the liquid nitrogen treatment, then you can mention what might be these things. Either it may be proline, either it may be sorbitol, either it may be mannitol, either it may be trehalose, either it may be abscisic acid. But this proline, sorbitol, mannitol, they are considered as osmolyte or osmoprotectant. I am writing this word. It is osmolyte or osmoprotectant. Osmoprotectant. Okay. Now, what are the different types of cryoprotectant? As I already told, that cryoprotectant means it should or it can able to protect the tissue from freezing damage. Protect the tissue from freezing damage. What does it mean? Inside the tissue, there should not have any ice formation. Or we can call one word, that is anti-freeze. Okay, so that they can able to lower the freezing temperature. Importantly, they will increase the viscosity, and most importantly, by applying this, they cannot destroy the cells, or rather prevent damaging the cells. Okay, so there are two potential source of cell damage during cryopreservation. First one is, they may have the chance that they may create large ice crystal, and if it is there, then the cell is going to die. Okay. So second one is, there will be chances that intracellular concentration of solutes increases, and if it is increases means the toxic level can arise. So before or during freezing, these two possibilities may be there. So to avoid from that, mostly there is use of cryoprotectants. Okay, this point is important. Mostly it asks in your exam. What are the different types of cryoprotectants used? So what might be them? One is glycerol, dimethyl sulfoxide, mannitol, propylene glycol. In some cases, ethylene is also being utilized. In some cases, ethylene can also be utilized. But among them, this one is the most popular cryoprotectant. In short, it is named as, in small or abbreviated form, it is known as DMSO. You can note down DMSO. DMSO. Let us discuss about this dimethyl sulfoxide. So this is excellent dry protectant. Important one is non-toxic. Another important feature, easily permeable, and most important is it is low molecular weight. And further, during recovery part, easily washable. So for that reason, DMSO can be referred while utilizing or while there is need of cryoprotectant. So it is being most important as per the cryoprotectants are concerned. So how much percentage is generally utilized? 5 to 10 percentage is preferable while go for the cryoprotectant. So this slide is important as per your MCQs are concerned. Either it will ask only one option or along with this and the D option will be all of the above. So you have to remember this. Why there is a need, then you have to go for this feature. Non-toxic. Let us say question asked that TMSO is an excellent cryoprotectant. Why? So A option will be like this, B option will be like this, C option will be like this, and D option will be all of the above. Likewise, such questions may ask in your exam. So during this cryoprotectants or this process, what are the three different steps by means of which we can prevent this plant material? So first one is vitrification. Here I have mentioned two photographs. Okay, this is one of the frozen kidney. This is vitrified kidney. By looking at these two, you can judge that this one having more survivability. The muscles were easily visualized, whereas this one is totally ice. And microscopically, you can view that several ice crystals were made, whereas in case of vitrified tissue, there is no such ice crystals structure. So to avoid from that, generally the vitrification is a must. Vitrification means slow in freezing process. Okay, that is required, or we can say that it is a process in which ice formation cannot take place. So during this cryopreservation, if we can surpass this condition, then the majority or the major chance that the vitrified muscles or any type of plant tissue can survive in a better way. Second one is cryoprotective dehydration. What it does, what does it mean? It means that whatever water content available in that tissue, it should be minimized. Okay, so that dehydration process is required. So for dehydration, mostly sugars, polyols, or in a sterile flow cabinet or over silica gel, those water is being removed. Okay, so once this dehydration can be going on, then what would happen? If dehydration reduces the amount of water and increases the osmotic pressure of the intracellular solution, which depresses its freezing temperature, and important one is it will promote the vitrification. To avoid from such water, this dehydration is a must. Okay, and for that reason, this dehydration is being mostly utilized. Okay. Next one is regarding the encapsulation and dehydration. Last class we have discussed about this one, synthetic seeds, isn't it? So mostly if you try to prevent such type of thing, then better you can go for encapsulate those plant material. And we have discussed this in the last class. That is calcium alginate beads can be prepared in a concentration of high concentration of sucrose, which can be considered as osmolyte for this one. And if it is being there, then maximum probability that the plant material must be preserved inside that encoded material. So this is the encapsulation method that we have discussed yesterday. Okay, once encapsulation occurs, the further one from here to here, let us discuss in the next part. Okay. So freezing condition, there are three ways of freezing. How you can freeze this. There is a rapid freezing process. Another one is slow freezing. We will discuss one by one. You just note down in your copy this point. This is important. This is asked in your exam point. This is important. Rapid freezing occurs at which temperature? Then you have to mention that it is minus 300 to minus 1000 degrees Celsius per minute. Okay, you just imagine per minute this must be there. Okay, why there is a requirement of rapid freezing? The quicker the freezing is done, this is important. Smaller the intracellular ice crystals are. So rapidly if you are freezing, then minimum chance that inside it there is chances of making any ice crystals. Okay, so in which category this rapid freezing can be utilized or preferred? These are the example: shoot tips of potato, strawberry, or brassicas. Mostly in these conditions they have utilized this. This has been shown how it is being uh, occurring in that process. Okay. Next one is slow freezing. Again, you have to remember this one. The temperature must be 0.1 to 10 degrees Celsius per minute. At times, this 10 may be given as 5 minutes. It's also okay. 0.1, it may be asking minus 0.5. It is okay. So, but this is the broad range, minus 1 to 10 degrees Celsius per minute. Here important is that this one is slow cooling. Slow cooling, what is the advantage of this one? Why there is requirement of slow cooling? The important is that whatever the flow of water from the cells available inside the cell, that can be taken out so that there is no chances of any ice crystal formation inside it. So for that reason, this can also be preferable. Here it has been showing two things. Okay, this is totally ice. Here you admire, this is there is no ice crystal inside it, isn't it? So the examples were the meristems of peas, then potato, then cassava, then strawberry. These are the examples where slow freezing were preferable. So for that reason, at times slow freezing is better than the rapid freezing. Depending upon the plant category, you can go for it. So this is the difference between these two, slow freezing and that of the fast freezing. What we have discussed, this is a gradual process. It will take times about 3 hours. Okay, there is less amount of dry protectants needed and greater chance of ice crystal formation. This is one of the negative feature. Okay, and in this process, mostly the eggs stored for fertilization is preferring slow freezing. Whereas fast freezing case, this is rapid one, as we have discussed. Within 5 to 10 minutes, it is the process is being over. Higher amounts of cryoprotectants are needed because you are incorporating the tissue in a rapid way. So for that reason, it must have higher coating material. This will not make any ice crystal formation inside the cells, very less chance. And during the eggs warming for fertilization, mostly this process is being utilized. So in a short, we can say that this is the difference between the slow freezing and that of the that is fast freezing. Okay. Next one, stepwise freezing. Those were the two different steps. Stepwise freezing means what it will do, as the name indicates, initially what they will do, the freezing temperature is being down to initially minus 22 degrees Celsius. Okay. After that, here it should be minus. After that, they will take a break of about 30 minutes. Then they will go for minus 196 degrees Celsius. Initially, we have marked that fast freezing condition, it was minus 300. The initial condition is minus 300 to minus 1000. But in this category, initially it is minus 20 to minus 40. After a gap of 30 minutes, they were going for the minus 196 degree. And this is the main part in case of cryopreservation. Okay. So if you are going for the slow freezing, then it permits the protective dehydration of the cells, but rapid freezing mostly prevent the growing of the ice crystals. Okay. So these many things occurring. So in a case, why we are going for slow freezing? Because we want to dehydrate the cells. Why we are going for rapid freezing? There is less possibility of ice formation inside the cells. Okay, and this technique is being utilized successfully in case of strawberry as well as in case of some suspension. So this is stepwise is generally preferable. Now come to the storage part. Storage part, once you perform all these things, it should be stored for a longer period. Okay, and this temperature must be maintained at that minus 196 Celsius. At times, maybe the range may be minus 70 to 190, minus 190. But the cryopreservation, the temperature is always or around the minus 196 degrees Celsius. So that this minus 196 degrees Celsius, they can able to stop all the metabolic processes, so that there is no question of any biochemical injury. These are the liquid nitrogen tanks in which the storage materials were being preserved. Okay. Examples I will show you where these liquid nitrogen containers are there. In Gujarat, also several places they have liquid nitrogen facility. Next procedure after storage, if you want that material or reuse, then you have to go for this process. You have to remember this word. Okay, remember this word. That means this is a reversal of the cryopreservation. That is removal of those preserved samples to the outside condition. So what they will do, whatever sample is placed in that appendrops or ampules, those were first they will have to remove that or put them at a temperature around 35 to 40 degrees Celsius in a warm water bath. Okay. Then what they will do after putting into warm water, there must have certain swirling action or shaking action must be there, so that you can just mark that the ice disappearance start to occur. You have to recognize that if ice were gradually removed, then this is the best one. Okay. Then once it is being done, then the tubes should not be left in the warm water bath after ice. Once this ice melt occurs, you should not put them further into the water bath. It should be transferred. Okay, it should be transferred to which one? To room temperature. Okay, this is a gradual process. First, you have put them into water bath. Once starting the ice disappearance, you have to keep them in room temperature. But the swirling action must be going on. That is 15 seconds for 15-second period. Then it will be go for now tissue which has been frozen by encapsulation or dehydration, that must be coming to a natural temperature or ambient temperature, which should not deteriorate the condition of the tissue. How it was going on, let us see. This is that condition. These are the ampules. This is the warm water pouring over here. Okay, so that the ice will be removed and it is being taken out. Okay. Now last stage is that once thawing over, how can we recognize that those cells were living or non-living. Naturally, few parts of that may be undergoing the killing action. So chances are that the death of the cells may occur. So how can we decide? For that reason, there are three techniques by means of which, or staining techniques were there, by means of which we can suggest that those were living or non-living. So there are three things. One is FDA test, fluorescein diacetate. You have to remember by heart. This question asked in your certain viva also, as well as this is MCQ. Very important. I am repeating. This one is meant for the living condition, living cells. This one is for dead cells, and this one also for living. But this one is the staining process. So let us discuss. These are the staining technique by means of it. Otherwise, you can count the cell number, dry weight, trace weight. Accordingly, you can also measure that the tissues which were cryopreserved are living or not living. So let us discuss these three techniques in details. So let us first discuss about the TTC. What is the full form of TTC? It is triphenyl. You have to remember the name, triphenyl tetrasodium chloride. So generally, if the cells are living in nature, then what would happen? They will make a product which is named as permagen. Okay, and it should be, the color should be red color. You have to remember this one also. Okay, so once these cells were added with TTC, the TTC gets reduced to make this red permagen, and this color can be measured spectrophotometrically so that we can say that these cells are surviving. Okay, so how can we determine which part of the cells actually stained with this red color? Mitochondrial dehydrogenase enzyme, which is found in mitochondria, actually it will take this red color. Okay, TTC, it will be converted into red color permanently. So you have to remember this enzyme also, mitochondrial dehydrogenase. And those dead cells, because cells are dead, there is no question of mitochondria, there is no question of this enzyme, then dead cell will not take up the gas. So the remaining cells, you can say that those cells are dead, whereas these cells which are red or margin, those were the living ones. Further, events blue staining. Here you have to remember the stain will make the cells blue. Okay, and which cells will take this stain? The non-viable or dead. So togetherly, you can say that if you apply that TTC or this one, later on I will show you a photograph by means of which you can understand. So how much is required? Only you have to apply 1 drop of 0.1% solution. This is very less amount. Easily you can put that and you can visualize the light microscope. Okay, so how can you determine that how much percentage of the viable cells are there? There is a formula called as number of fluorescent cells upon total number of cells. Total means how many are viable and how many are non-viable into 100. Now question comes, why there is need of this, or how can you determine these fluorescent cells? This can be done by FDA. FDA, that is fluorescein diacetate. Here you mark this green part or fluorescent part, those were the survival one, whereas the rest part which are you are marking, these parts or these blue dots, those were events. Okay, events. So looking at that, you can say that these are the dead one, whereas these are the living green one. So fluorescein diacetate make the compound or make the cells into fluorescent one. It will produce or appearance like the green, and accordingly you can say that this is dead or which is living. Now come to the applications. Why we are going for it, or what are the benefits? The first and foremost thing is that, as we have already discussed in the definition, we want conservation of this genetic material. In the last class also, I have told you that to protect these endangered medicinal plants, if we can preserve those samples, then we can chance that we can go for a longer period of time. So, what might be the plant materials? Meristem, anthers or pollens or embryos can be conserved. Second one is we can store the cell culture in freezing conditions. If you want to maintain this cell line or subculture further, initially you have started this time, after 6 months, after 1 year, if you want that, you can preserve it under the liquid nitrogen condition. Okay, why? Because they will suppress the cell division, so that further modification can be done after the thawing process. Third one is, what might be the third application? The maintenance of disease-free stock. This is one of the important one. While you are choosing any plant material, if it is disease-free or pathogen-free, those plants should be frozen or further used, and that's why that can be also preferred. Fourth one is cold acclimatization and frost resistance. Actually, acclimatization means those tissue or those plants which are accustomed to such type of thing. That means they can survive in that condition. Those were preferable. Okay, let us say this is X plant, this is Y plant, this is Z plant. Among them, let us say Z is generally the frost resistance or any type of resistance capacity they have. Then what would happen? That can be preserved in cryopreservation technique, and it can be utilized further or whenever required we can utilize it. For that reason, this is also one of the important applications. Now come to the related topics, that is seed bank. So seed bank, as the name indicates, mostly the seeds were preserved here. Okay, this is also a type of bank. Okay, so in this category, what might be the different thing? The seeds may be stored, maybe either it may be food crops or certain rare species. Okay, and mostly the seeds are dried to a moisture content less than 5%. It is not related to cryopreservation, but it is respective part. That means that is bank related. Okay, we want to store that. You just look here, it can be stored at minus 18 degrees Celsius or below. It is too much higher than the liquid nitrogen. Okay, this is the some examples where it is being stored. This is seed bank. You can collect. Another one is gene bank, where the genetic materials are being preserved. Okay, what might be the different genetic material? Let us say in case of plant, there might have certain freezing codes. Some of the seeds were also being preserved. Okay, or if you want, the freezing sperm can be also being preserved, or certain eggs wherever required. Mostly in the zoological freezers, these were preferable in case of gene bank. And one of the best example is 12 god global seed vault. This is the best example of gene bank. This is that pictorial form of this one. They were preserved this. Okay, and these are the different examples. National Seed Storage Laboratory, which is found in Colorado. Mostly 2100 accessions of apple were stored. Another example is National Clonal Germplasm Repository, that is also found in USA. Potato Center, International Potato Center, that is found in Peru. You just mark here 345 varieties were preserved over here. tissue culture BC research inclusive that is found in Canada. There are about 14 conifer species, about 5,000 accessories were stored over there. And these are certain other examples of this FO cell, that is Association Forate Cellulose, that is found in France. This is another one, National Institute of Agrobiological Resource of Japan. We just marked about 50 accessions of mulberry were preserved over there. These are certain examples stored in that respective uh, banks. IRD is another one is found where you can find out there are 80 accessions of oil palm were there. Another one is German collection of microorganisms and cell cultures. There are about 519 old potatoes. These are backdated some examples. Now more may be added to it. Okay, banana accessions were also being stored. But what is the main problem with plant cryopreservation? To think that there might have certain problem, yes. Okay, cryopreservations mostly available at different plant species, 150 to 200 has been added to this cryopreservation technique. Okay, it may be more than that also. Okay, but what would happen if this cryopreservation protocol, if it is being not perfectly processed or followed, then there might have certain problem created. Okay, so it is being empirically adopted, or you can say they're standardized. Okay, so depending upon their natural freezing resistance, let us say you have taken the X species, it requires certain natural freezing resistance capacity. Y will be different. So accordingly, you have to be standardized before going for that. Secondly, you have to be cautious about their explant size and the type of the explant. In some cases, if you are going for the nodal culture, in other case, tube tip can be preferable likewise. And also, you have to be cautious that how much water is available in that test so that during the dehydration process, it should be answerable. Okay. So for that reason, mostly during the academic studies, it is being utilized. Okay. And you can say that only few plant germplasm collections stored in liquid nitrogen, not all. Okay, very limited amount. Because for that reason, you have to be also maintain such type of liquid nitrogen storage. Okay, so and also you can say that there is no real standardized methods available. In some plants, you can prefer the different dry protectants. In some cases, different. For that reason, you have to make a standardized protocol for your need whenever it is required. Okay, but why this cryopreservation is not more widely accepted? Because it is not available with so much of desire. In some cases, what would happen during grinding process, when you try to cross the tissue, at that time also liquid nitrogen is required. But during liquid nitrogen handling, it should be very cautious because it is -1. Let us say if during your use, if a drop of liquid nitrogen fall upon your hand, then it may be broken down. The tissue may damage. Okay, because you have not utilized dry protectants in your hand. So for that reason, this is also being a dangerous prospect while utilizing this process. No doubt you have to use it while using, you have to be cautious enough. Okay, then it is not so much aware about this. No, everyone is not uh, aware of it. Okay, for that reason, this process must be learned by the plant researchers so that it can be further being popular. Okay. Then in some cases, lack of coordinated research. Let us say if you want to cross the plant sample by applying liquid nitrogen, if it is not available, what you will do? You have to go for any alternative. If it is a must, And if it is available in all the institute, then definitely you can go for it. Okay. So the cost action like CryoPlanet could make the difference. If the cost will be reduced further, then it can be referable. Okay. These are the references which you can follow, and some review articles are also there for cryopreservation. So you can follow that. So these are the reference. You can go for it. Okay. Any plant biotechnology book also having this cryopreservation technique. So this is all about cryopreservation.